It's nice to see everyone. This is now the third introduction I've given this year. So we'll start at the beginning. My pleasure to be here to introduce Dylan MacArthur-Waltz from his I wrote a few notes. Dylan grew up in Missoula, Montana, the son of an architect and a psychology professor. Psychology professor is here today. He still loves Montana to this day and returns whenever he can to be with his family and to spend time tromping around the mountains. Dylan did his undergraduate at Stanford University, where he worked with several notable folks, including Deborah Gordon, Aaron Mordecai, and others that you would know. Remarkably, he is a co-author on 5 excellent papers. I had to count them again. 5 papers from his time as an undergrad, including a first authored paper about temporal activity patterns in ants that he did with Deborah Gordon in the Journal of Animal Behavior, and a PNAS paper about biodiversity and tourism in Costa Rica. So when Dylan visited UC Davis as a prospective student, I had already read his application and I had read his glowing letters of reference, and I already knew that he was capable of extraordinary things. But what I was most impressed with during that visit and henceforth has been his humility, his earnestness, and his self-effacing sense of humor. During that visit, it was notable, he never once tried to say what he thought I might want to hear. He just said what was on his mind. He wasn't afraid to tell me that he wasn't really sure about his long-term goals. And he was quick to have a chuckle about past field experiences that he didn't really enjoy. He did mention that he hoped he could someday do work with conservation value. And maybe without ever really trying, I think he understood the most important rule, which is know thyself. So it was no surprise to me that he hit the ground running in grad school. He always found ways to be productive and creative, even while surrounded by a global pandemic. He designed ambitious field experiments, lab experiments, and data projects. He was open to all possibilities and was really starting to hit his stride at the end of his 3rd year. And then on May 28th, 2023, I received the following Slack message. Hey, Louie. I had a pretty scary chain of events yesterday. I had a seizure and ended up in the ER, where I learned that it had been caused by a brain tumor. Chances seem quite good that it can be removed, but I'll be trying to move quickly to get that done in the next few weeks. I think I'm going to need to step back from lab stuff for quite a bit in the meantime. It has been 1,011 days since that scary chain of events. And in that time, Dylan has courageously walked down roads of human experience that I can scarcely imagine. He had a tumor removed from his brain, a process which he later cheerfully described as something akin to removing a cherry from a bowl of Jell-O. He has had to learn more about human neurobiology than most ecologists will ever need to know. He has spent more time inside an MRI machine than anyone should really have to. And the MRI scans confirm that he has a clean bill of health, but he'll be getting at least 2 scans a year for the rest of his life. I once asked Dylan how many more times he would need to go in for an MRI scan, and he cheerfully replied, hopefully about 100. So we are here today to mark milestones, the completion of his PhD for sure. He has done genuinely remarkable work, and I'll let him tell you about that. But we're also here to mark 1,011 days well spent, and perhaps to appreciate each one a little bit more, and also to say thanks to all the people who've supported Dylan through the last few years. Also, Dylan studies butterflies.
Thank you so much, Lily. That was lovely. And it's so amazing to see all of you here today. This is like so many years of my life right here. I'm so happy to see it. I'm really excited to present this work to you all. So I'm going to be sharing my three thesis chapters, which, like Louis said, they're about butterflies. They're about milkweed-monarch interactions in the context of climate change. But before I jump into that, I want to take a moment to do a few acknowledgements myself. So acknowledgements. So first of all, thank you, Louis. This whole process has been possible because of you. I want to thank Louis for constant, consistent, and attentive mentorship throughout my time in grad school. Because of Louis, I am a more patient and careful scientist, and I've learned ways to use all the tools at my disposal to chase after questions that I'm excited about. And as Louis mentioned, I've dealt with a lot of health issues during my PhD, and his flexibility and support while I was navigating those issues is a big part of why I'm standing here today, finishing in the timeline I'd hoped to finish with some really exciting projects to share with people. So thank you, Louis. I'd like to say thank you to all the lab members, past and present. A bunch of you are here today, which is great. The Yang Lab has been a source of great friends and also just a really exciting intellectual community for my time in grad school. I'd like to thank my dissertation committee members, Elizabeth, Emily, for their guidance on my chapters and my career and my path through science in general. And finally, I want to just mention my co-authors in these three chapters who were instrumental to all this work. And I would like to extend a thank you to the PopBio community at large, all of the PopBio program, my cohort, and my friends in PopBio have really been like sort of the beating heartbeat of this program. You all are amazing. Grad school friends are really important. And finally, I'd like to thank my family, my parents. My mom's here today. And I'd like to thank my partner, Emily, who's been with me through the entire PhD, stuck with me through the whole cancer treatment process. She's absolutely the reason that I was able to do this whole dissertation. And finally, I'd like to just say a quick thank you to the amazing natural ecosystems of California and all the cool critters who live around here. This has been like a constant source of inspiration and just remembering why we do ecology for me in this program. So one of those particular creatures that's been really important to me during my dissertation is the Western monarch, shown here doing this amazing behavior where they hang in clusters from the branches of a eucalyptus tree. Monarchs are a really great insect system for looking at the effects of climate change because they're very visible, they attract a lot of public interest and attention, and they have a really unique and complicated life cycle that makes them vulnerable to a bunch of different climate threats. So to walk through that a little bit, this map is showing the Western monarch population, which I'll be focusing on today. So it's a distinct population from eastern migration, which is what a lot of folks are more familiar with. The Western monarchs spend their winters on the California coast, overwintering in groves of trees, often eucalyptus trees, where they find a pretty consistent, cool climate to spend the winter without a lot of activity. And in the spring, when it begins warming, they migrate inland to look for milkweed plants, their obligate host plants to lay eggs and breed on. They continue migrating inland throughout the summer, and then in the fall, they return to overwintering groves. So this, all these life history events require synchrony between climate and different locations, between different species of milkweeds and monarchs, and they're moving across sort of vast spatial scales. So there's a lot of opportunities for climate change to mess up this life cycle. And unfortunately, that does seem to be happening with Western monarchs. So this is a graph of the estimated abundance of Western monarchs. This is calculated every winter at couple wintering sites. So you can see that there's been a pretty tremendous decline in Western monarch abundance since 1980. And even in that little inset graph, there's been a pretty tremendous decline in the last 5 years as well. And this has gotten to the point where in these last couple of winters, including this past winter, unfortunately, we've had only a few thousand monarchs at these overwintering groves along the California coast, when we estimate that there used to be tens or hundreds of millions So things have changed pretty dramatically. And it is over this time scale that sort of correlates with the scale of a lot of climate change. And there are certainly a lot of other possible explanations for this population decline. Some important ones are habitat loss, pesticides, possible sort of disease ecology dynamics. But I'm going to be focusing today on effects basically connected to climate change directly, so temperature and precipitation changes, among others. So the chapters I'm going to walk you through today, these are 3 chapters in my dissertation, and I'm going to walk through them in the order that a monarch might experience in their life cycle. So first, we're going to start with where monarchs begin the year at coastal overwintering sites. And sort of the first phenological event that these monarchs undergo is the decision of when to depart those overwintering sites and begin their migration inward. And the next chapter I'm going to talk about is going to explore what kind of environment do those monarchs find when they migrate. Specifically, is their obligate host milkweed going to have emerged yet? Will they be able to start breeding? Or will they be stuck in this sort of vulnerable period where the population is very low? They're not sort of safely ensconced in wintering sites, and they're not able to breed yet. And then finally, I'm going to wrap up thinking about the experience of later generations of monarchs spending the summer in inland areas, the California Central Valley, Oregon, Washington, Utah, places that are increasingly experiencing extreme heat in the summer, and think about if that extreme heat may directly cause mortality to monarch eggs and caterpillars, and if this could be an issue for populations. So I'm going to jump in with my first chapter on leaving overwintering sites. So this is this first phenological decision monarchs make as they're beginning their yearly life cycle. And there have been observations from folks on the ground who pay attention to these clusters that they seem to be disaggregating earlier in the spring than they used to. So this is a process that we used to think of as happening in March, perhaps, and now we're thinking of it happening in February. But we haven't fully understood why this might be happening. And for my dissertation, I wanted to investigate, could this be an effect of climate change? Could this be tied to increasing temperatures at overwintering sites? And there are some reasons to think that this might be the case. So monarchs, overwintering monarchs, are in a sort of hibernation-like state, and they rely at least partially on fat deposits from the previous summer to survive winter. And because they're ectotherms, their metabolism is tied directly to temperature. So the warmer it is, basically, the faster they're eating up those fat stores, and potentially like running out of food faster earlier in the year. And also, because they're cold-blooded, they also just can't fly when it's cold. So there are sort of like direct reasons that if it's warmer, they're probably flying earlier. So to sort of step into the data for this project a little bit, we're going to be focusing on overwintering sites along the California coast and a little bit down into Baja California. There are over 500 known or suspected overwintering sites in this area. They're mostly coastal, and it's thought to be that they're so coastal because monarchs are searching for this cool, consistent climate where they will not, they can sort of like put themselves in a refrigerator. Like they can sort of preserve these fat stores, not burn through them too fast, but also not encounter freezing temperatures or sort of dangerous temperatures. So, although there's this huge range of overwintering sites, the most significant sites are in this sort of central California area between about Santa Cruz and Santa Barbara. And to get a little sense of what these sites look like, this is a picture of the Pacific Grove Monarch Butterfly Sanctuary. So they're right along the coast, usually in these big groves of trees. And I guess it might seem a little silly to ask the question of have these sites been warming, because we're also used to the assumption that everything has been warming over the last 40 years. But these coastal sites do have a lot of complicated thermal dynamics. They have onshore winds, offshore winds. Sea temperatures are really important. So it seemed like an important first step for this project is to make sure, are these sites actually warming? So this is looking at the change in average winter temperature over the last 40 years for all of those 500 sites. And the takeaway from this is that, yes, it does seem like over the past 40 years, these sites have warmed by about 0.02 degrees per year. So that's about a degree of warming since 1981 on average. And I was curious to see if this trend extended past the 1980s. So if we could get sort of a longer term picture of the climate at these locations. And in order to work on that, I paired contemporary measurements of temperatures at overwintering sites with long-term climate weather station data that goes back into the early 1900s. And using a quantile model, I was able to extrapolate temperatures at overwintering sites back to the early 1900s, even the late 1800s. So pretty interesting to see these sort of really long-term climate trends at these sites. And there does seem to be some more complexity than sort of simple warming, but in general, it does seem that these sites have warmed over the past 100 years. So having sort of established that these sites seem to be warming, I wanted to try to figure out what effect will that have on when monarchs leave their overwintering sites. So the data that I went after for this were data that would allow me to sort of quantitatively measure departure time of overwintering groves, with the goal of using yearly and site-to-site variation in temperature to study the effects of temperature on disaggregation. So the data that I ended up looking at looked like this. They're data on seasonal abundance, and particularly I'm interested in the right-hand side of that curve, where monarchs are departing from their overwintering sites. So this is an example here of data here from Pacific Grove in 2017 and 2018. And this project, to me, is a real testament to the importance of citizen science and the strength of citizen science data, because folks are so invested in these sites that people have been out looking at monarchs and counting monarchs for decades. And this means people on the ground with binoculars trying to count monarchs and estimate monarchs' atrial branches. It's pretty amazing. So I never could have collected a long-term data set with enough observations to do this project myself. But thankfully, a lot of people have published these data or were willing to share it when I asked them. And I ended up with a data set of 70 site seasons encompassing 20 sites, 23 different sources, and about the last 25 years. So thank you to everyone who helped collect these data. Just a quick shout out to all the sources. And then I moved on to thinking about how would I quantify these data and try to get a qualitative metric of when monarchs leave. So I first started by fitting the lowest fit to these counts, which are often quite messy. Used the lowest fit to estimate the max value, which is shown by the red dot here. And then I used that value to calculate metrics, including the percent of the max count remaining on January 31st. So this is like a kind of a retention metric. I calculated the rate of decrease between the maximum and January 31st. So that's sort of a rate of decline metric. And then I also calculated the date of the year when 50% of the max remained. And then I regressed all of these against mean temperature. And importantly, all of these metrics should be somewhat independent of the overall number of butterflies in the site. So it's not just going to be showing like were there a lot there or were there not a lot there. It's going to say like when are they moving. So jumping right into those data, this is showing those three metrics that I just described regressed against mean temperature. These are partial effects plots. And the results of this analysis show pretty clearly that warmer winters do seem to cause earlier disaggregation. So if the average temperature in winter is higher, fewer of the max count of monarchs remain. The loss rate is higher. And the day of year when 50% remain is earlier, if it's warmer. So this seems to support the sort of first hypothesis I had with this section, and does seem to indicate that there could be a climate change connection to disaggregation timing. And we also found a sort of interesting effect from this analysis that I was not anticipating necessarily, which was an effect of cluster size, so the number of butterflies at the overwintering site on departure time. So what we found is actually a pretty strong effect that larger clusters tend to stay together for longer. And these are proportional metrics. It's not just saying if there were more in January, there's more effect. It's saying if there's more in January, a larger percent of that what's there is left. So this is pretty interesting. Definitely still kind of thinking about this, but there is some evidence in the literature that these larger sites tend to attract butterflies from nearby sites, possibly. So it may be sort of a cross-site aggregation process that's driving some of this trend. So this was really cool. It's exciting to see these results. And I sort of had one more little piece that I was hoping to put together for this chapter, which was, okay, so we see that there's this pretty clear temperature effect. We see that these sites have been warming over the past 100 years. Are there any sources of information about overwintering disaggregation that could go back 100 years? Could we actually see this happen? And it turns out that because these sites have been of interest to humans for so long and have been tourist destinations for the past 100 years, there have been quite a few newspaper articles written about them. And often these articles include information about when to visit them, which includes information on when they leave, but when can you not see them anymore. So I started looking into some of these historical articles and found a bunch of these from the early 1900s. This one has a quote, this kind of amazing quote. By the middle of March, there is no longer any need of shelter, and the colony begins to break up. By the 1st of April, they have entirely disappeared. And this is information from 1921, which is just so cool. So I started looking around to see if there might be enough of these articles to actually do a quantitative analysis, and ended up with a bunch of help looking at over 5,500 newspaper articles mentioning Western monarch butterflies. And I split these articles into two time periods: a historic time period from 1900 to 1950, and a contemporary time period for comparison from 2000 to 2025. And after examining all these articles, we found 30 historic and 38 modern articles that seem to have their own separate information about when monarchs leave overwintering sites. So looking quick, and then we trickiest part of this analysis was using those written descriptions to actually generate a date. So we created a sort of complex rubric for trying to assign each written date an actual calendar date, and that resulted in these data here. So the historic data are shown in red on the left, modern data in blue on the right, and then our best estimated departure date on the y-axis. And what we found from this was very much in line with what we found from the other two parts, that historic newspaper articles do seem to report that monarchs stick around longer into the spring than modern articles. And this difference seems to be on average a difference of about 20 days. So this was really cool. And it was a fun analysis to sort of be able to close the loop in those other two charts. So yeah, sort of moving on, it seems like from this first chapter, we have a pretty good idea that monarchs are leaving earlier than they used to. This is a strong effect. It's driven at least in part by temperature, and it's over this really long time period, about 100 years. So sort of armed with this knowledge, I wanted to move on to investigate what sort of seasonal landscape are those monarchs emerging into. Does it matter if they leave sooner or later? Sort of what are they finding is the most important thing. And in particular, milkweed phenology in the spring is likely to be a really important piece of this puzzle. So, we can see in this timeline here, this is sort of from chapter 1, that monarchs are leaving sooner in the modern era than in the historical era, which could potentially mean that this length of time between departing and having milkweed available is longer. But there's sort of an assumption embedded in that, which is that the milkweed timing has not changed over that same period of time. So that was the assumption that I was hoping to dig into in this chapter. So the data set that I used for this chapter was a database of about 13,000 observations of milkweed from the app iNaturalist. Milkweed are great with iNaturalist because they're big, they're showy, they're easy to identify. Lots of people upload them. So this is a really unique data set that covers, most of the range of these species, covers many species. Thanks to everyone who uses iNaturalist. I always say that. So from these observations, I was able to extract phenology information about the stage of the milkweed, like flowering in pods, like we see around the room. The location of the observation and the date of year and the species. And I paired all that information with the spring temperature that that individual experienced and the precipitation that that individual experienced as well to see if those climate factors change the date of year of observation. And the basic answer here, well, the first answer that I found was milkweed phenology changes a lot from year to year. So this is just showing across all of the data for these 5 different native California species, the average day of year of 3 phenological stages, budding, flowering. And there's a lot of year-to-year variation in these trends for all species, for a bunch of life stages. That variation is on the scale of about 2 to 3 weeks, which is interesting because we're looking at sort of the same scale as the variation in overwintering departure. And then I moved on to asking how much of that variation could be attributed to climate, in particular to spring temperature and rain year precipitation. So there's a ton of interesting stuff going on here. This is showing effect sizes for a bunch of models, phenology models. I have, this stuff is all published in Journal of Ecology. So I've talked a lot more in that article about sort of species-specific trends and life stage-specific trends, if anyone's interested in that. But for this talk, the main takeaway is temperature, all these coefficients are negative. So if it's a warmer spring, milkweed emerge earlier. Precipitation, all these coefficients are positive. So if it's a wetter winter, milkweed emerge later. And I will just quickly sort of hint at this. We were also able to go even further with this analysis and look at local adaptation in phenology responses to climate, which was a really cool thing to add on to this analysis because we did not have repeated observations of any individuals. So this was just very cool that we were able to do this. And again, more details in the paper. The sort of like short version is that we did find some evidence of counter gradient local adaptation in these phenological responses where essentially plants at hot sites are less sensitive to heat and plants at wet sites are less sensitive to precipitation. But back to sort of this main question, this main result of warmer and drier conditions leading to earlier phenology does actually seem to hint that changes in milkweed phenology could potentially help offset early overwintering departure. These effects are both in the same direction, so warmer is earlier, and the magnitude seems like it could be reasonably similar. But there are still a ton of questions left about this. Like probably the biggest one for me is coastal and inland temperatures, are they actually tightly correlated? Also, it's unclear sort of how plastic, like what the limits of plasticity are on each of these traits as the climate continues to warm. So maybe a future paper. This is where I'm going to leave it for now. So this was our sort of first 2 chapters here, thinking about this first spring interaction of departing overwintering sites and then trying to find the first milkweeds to reproduce on. And so moving on to the 3rd chapter here, after monarchs have left these overwintering sites, they've made it to some early spring milkweed. We're starting to see those summer generations happening where milkweeds are expanding into the sort of central western US. And they're going through multiple breeding generations, and hoping that the population is growing really fast at this point. But climate change does pose another threat at this point in the life cycle, which is extreme heat. So here in Davis, we know all about extreme summer heat. And it makes sense that if you had to sit outside in that temperature all day, it might be pretty bad. And extreme heat is a problem, not just in Davis, obviously, but lots of parts of the interior West get really hot. So for this 3rd chapter, question was pretty simple. Basically, do monarchs in the field die from extreme heat? And how hot is extreme? So how hot does it get does it have to get before monarchs just start dying because literally it's too hot? And I'm looking specifically at eggs and larvae for this part of my chapter because adults can leave, but eggs and larvae are kind of stuck. So we had a little bit of context for this chapter from literature, which was really helpful. So in the lab, folks have done a lot of experiments of exposing monarch eggs and caterpillars to high temperatures. And we have some results, like the fact that continuous exposure to 91 degrees Fahrenheit is lethal for most eggs and caterpillars. But these results don't always translate super well to the field, because in the field, monarchs have other strategies to avoid heat stress that they can't access in the lab. So for one of the most important things is that monarchs can actually recover during cool nights. So if night gets cooler, monarchs can get up to about 97 degrees and seem okay. They also can change color to get lighter if it's hot. It's pretty cool. And they can seek shade as well to stay. So the goal with this project was to sort of bridge these this sort of small scale lab studies with this information about the field and try to investigate this question about extreme heat stress at a continental scale. So individual field studies are very context dependent. Lab studies don't capture the complexity of the field. And this is where this monarch larval monitoring project data came in. So this is yet another citizen science data set of folks who are really excited about monarchs and go out in their own backyard or their own community to survey for monarchs at a patch of milkweed, usually about once a week. And this is a super cool project. It's been going on for about 20 years. And the sort of size and extent of this data set allowed us to try to use it to look at the effects of exposure to hot temperatures in survival. So to look a little bit at what those data look like. So we have people doing repeated surveys at the same locations looking for monarchs. And we paired a survey on day 1 with a survey on day 7 into what we call the survey interval. So it's sort of a before and after measurement. And the idea was we can look at the eggs and small caterpillars present on day 1. And if they survive, they should be present as larger caterpillars on day 7 or 8. And possibly, if they've experienced cooler temperatures during this interval, we'll see high survival. So most of the ones we see at the beginning will have survived. But if they've experienced high temperatures, possibly we'll see lower survival. So this is seems very simple, but it was not very simple once we started working on it. And we ended up having to develop a pretty complicated modeling framework to try to deal with a bunch of challenges with these, both with the structure of these data and also just the biology of the system. So one challenge is that monarchs grow faster when it's hot. So we had to make sure that we were accounting for that and not assuming that caterpillars had died when they had actually pupated. And this model was also super zero inflated, so we had to struggle with that. And we ended up with this modeling framework that is modeling the number of survivors as a function of both the thermal stress accumulated during the interval and the number of beginners at the start of the interval. We use this zero inflated negative binomial GAM structure, and we included offsets for site size. Sites vary a lot in their size, and also for the length of time between the surveys in case they were all 1 week. And we also have included a spatial smooth to try to account for any spatial information. So the end result of this model is that we've been able to look at the effect of thermal stress degree days accumulated during that interval on the expected number of survivors at the end of an average interval. And we chose to investigate the effects of different thermal stress thresholds to try to get at that question of how hot is too hot. And is it only temperatures above 36 that are really damaging? Is it any even moderately high temperatures causing a hit to survival? And this is what those data look like. So you can see, as you might expect, that the expected number of surviving monarchs decreases as more thermal heat stress is accumulated. So So this is showing that yes, heat stress is causing mortality. And you can see, looking at the difference between these different model thresholds here, that basically the higher the heat stress, the faster we see this. So this was somewhat surprising to me to see such a strong effect of even temperatures above like 30 degrees Celsius, which we had not really thought were that stressful to monarchs previously, but also to see these really quick effects of high mortality on monarchs at higher temperatures. So another way to sort of look at those results that's maybe even a little more intuitive is looking at per degree day and per hour mortality. So a degree day above 30 degrees Celsius would be 24 hours at 31 degrees Celsius or 12 hours at 32 degrees Celsius. It's sort of this linear accumulation. And we can see that so one full day at 31 degrees Celsius is already causing about 20% mortality, which was pretty surprising to me. And we can see by the time we spent a whole day at 39 degrees Celsius, we're seeing really, really high, almost complete. So this is kind of sobering, but seems like definitely a suggestion that summer extreme heat in the summer may directly be a threat to monarch populations. Some summer habitat that we've been sort of counting on may be becoming lethally hot. So hopefully this can sort of empower us a little bit more to think about what suitable habitat would be in the future. So yeah, I wanted to wrap up with thinking a little bit about the sort of conservation angle, like Louis mentioned, that's been an interest of mine throughout this program. And I'm hopeful that some of this information may be useful to leverage to try to help the sort of Western monarchs. So in particular, in chapters 1 and 2, I've thought a lot about this important time period between leaving overwintering sites and finding milk in the spring. And I think a lesson that a lot of folks are sort of taking from that right now is that focusing on those early season species that emerge and grow really early in the spring may be especially impactful, making sure that monarchs can breed as soon as they move overwintering sites. And it seems possible that even focusing that restoration in hot and dry areas might be especially helpful. And for my extreme heat mortality chapter, there is this sort of sobering side where it's like, well, we can't really mitigate heat directly, but we can think about trying to focus habitat restoration on areas that aren't going to get lethally hot. And we can think about prioritizing shade at the top of the movement. This is another way to give a little bit more buffer. So yeah, this is my thesis. Really excited to share with you all. I wanted to just quickly tell you what's up for me now. So I actually graduated in December, and I have been working since then as a postdoc with Sydney Record at the University of Maine. And we are working on a project using neon plant phenology data to try to look at the drivers of community level flowering synchrony across all of the 47 neon sites. So this has been really fun. Just gotten started on that in the last couple of months. I am working remote, so I'm still living in California. I'm living in Northern California in Humboldt right now and moving to LA in August. So I'd love to see anyone in either of those places. It's very remote up there. So yeah, thank you, everyone. Yes, Elizabeth.
So it was really fun to see. I love the way you put all that together. And I'll say randomly that I realized we may have a more direct Montana connection than I realized from my days in Missoula, but we'll talk about that later. But I'm wondering in your last chapter, first I want to compliment you on it because I've seen other people try to get something out of those volunteer collected data for survival and not succeed. But my question is, people worry a lot about heat and survival of monarch larvae in the summer, but monarchs also live year-round in places like Costa Rica and Brisbane, Australia, I think, and Hawaii. And so how does this climate compare to that climate, and why are we worried about heat?
Yeah, that's a great question. I think something I would pull in right away is the humidity angle. I think that's something that we weren't really fully able to develop with this data set. But especially in the West, I think that desiccation is probably a big risk. And so this high temperature may be acting through desiccation in a way that might not be Hawaiian. So I think the humidity angle is definitely really important. And I think part of this chapter was just driven by what folks have been seeing in the field. Some of these summers have been really, really hot in the late summer. We've seen sort of a promising population throughout the spring and early summer, and then this sort of population crash around the time that it gets really hot. So I think there's definitely, reasons to think this might be. Yeah, Rick?
If I understood you correctly, in the last chapter, you were sort of lumping eggs and first instars, and can you separate those? And would you expect that, like eggs and neonates and later early instars would, quite different responses to thermal stress?
Yeah, that's a great question. It is possible to separate those stages in this data set. We chose not to because the information I could find in the literature about thermal tolerances seems to be within the same sort of general area for those different life stages. Interestingly, eggs are often more tolerant than some of those other stages. So there could be interesting stage-specific differences, but we haven't tried to piece that out. And I think the general result, transferable. Yeah. Here.
Yeah, that was a really awesome talk. Going to your first chapter, I had a question about, I was really interested in how you were saying that cluster size had a huge impact on how quickly they were, leaving and how soon they were leaving. And I was wondering, given how large the population decline of monarchs has been in the past, 50 or so years, do you need, I'm sure you've thought about it, but do you need temperature to explain the earlier thing? Or could it just be the population's, it's a density dependent factor that's, driving this?
Probably. I think that's a great question. That would be my next step. I haven't done that yet. But I think it would be really cool. I think we do have the data here to sort of close that circle as well. How much, that 1 degree change in temperature, how much should that advance departure? And that reduction, orders and orders of magnitude, how much should that advance? My guess would be about long-term change.
Awesome, thank you.
Yeah.
Back to the summer heat story. Do the monarchs show any evidence of behavioral adaptation? Is there a chance that they can choose new spots?
Yeah, that's a great question. So eggs don't have a lot of behavioral adaptation. Caterpillars have a couple options: they can change color, they can do some. I think maybe what you're hinting at is a question of like, can adult female monarchs move to cooler areas to lay eggs? And I think that's a great question. I'm not sure we actually know that, but that would be a really interesting question. I guess it might emphasize the importance of heat waves, especially because if adult females are able to choose cooler areas, they would probably not be able to anticipate a heat wave, would be my guess. And then that might be when we see the highest weekend. There.
That was so good. So still on this one, do you think that predation on these guys is are there more or less predators that are hotter or colder temperature? Like, do you think they're getting eaten or just dying? Or dying and then getting eaten, I guess?
That's such a good question. We've thought a lot about predation, and it might be really effective. And the main predators of monarch eggs are insects. So they're also cold-blooded. They're probably also sort of moving faster at higher temperatures. So it's sort of complicated because monarchs develop faster when it's hotter, so they're escaping predation more quickly. And also the predators are maybe also searching more quickly. So there's a lot of sort of things about there, but my guess is that predation is not like the main driving factor. but absolutely throwing that out. It would be great to actually do that experiment.
All right, let's thank Dylan.